PCR, or polymerase chain reaction, allows us to make lots and lots of copies of defined sequences of DNA in these teeny tiny little test tubes using this machine called a thermal cycler. What it's going to do is it's going to start at a high temperature to like melt the two strands of DNA apart, so like unzip the strands. Then it lowers the temperature a little. You have this anneal step where basically you're going to dictate where you want the sequence to start and stop on your template DNA. So it can't make a sequence from scratch. You have to give it like a template, and so it's going to melt the strands apart. So at a high temperature, then lower the temperature to allow your primers to bind. So primers or like oligonucleotides are like these short pieces of DNA that are going to bookend the regions you want to copy. Those are going to bind. Then you're going to raise the temperature a little, and you're going to have an extension step where basically this enzyme, this reaction helper upper that you have in there, DNA polymerase, it's going to use the one strand as a template, and starting at that primer, it's going to copy the strand. So in the first cycle, it's kind of just going to like go till it runs out of steam. But then you can keep cycling this. So if you remelt the strands, and now you use those copied strands as template strands, now what's going to happen is that when you run off, instead of running out of steam, you're going to run off the end, and the end that you run off is going to be where the start of the other strand was, so where the primer was for the other strand. And so this is how you can use primers, one per strand, in order to bookend the regions that you want copied. So let's take a look in more detail. So let me introduce you to an analogy that I like to use when thinking of PCR. I think about a polymerase chain reaction instead of a polymerase chain reaction. And basically, we can think of DNA as being our like train tracks, and then DNA polymerase, so the copier is going to be like our train. And this train is going to copy a sequence of DNA between like two train stations. And so there's some unique things about this train that are going to dictate how we're going to set up the reaction. So we have our template, which is the part of the template DNA was like the whole transcontinental railroad, and then we're only wanting to copy like a section of it, say. So our DNA polymerase can travel between any two cities and lay down track ahead of it as it goes, but it can only travel along a half track, so like a single-stranded track. So we can heat the DNA up to unzip the strands, and now it has these half tracks, but it doesn't know where to start. And so we're going to tell it where to start by adding these primers, these short pieces of DNA that are going to kind of allow it, they're going to form these start stations. So at the molecular level, basically this DNA polymerase, it needs a double-stranded region to start, but then it's going to travel along a single-stranded region, and it's going to use the track ahead of it as a template for laying down the tracks, the complementary tracks ahead of it as it goes. And then this will do it over and over and over again to make lots and lots of copies of this region. So one of the key things you'll see is that we have one train per track, like one train per half track. So one train per strand, one DNA polymerase per strand. We need one primer per strand. The reason for this is because these strands are anti-parallel. So these strands, one of these is running 5' to 3', and one of these is running 3' to 5. One of these is running 5' to 3', and the other is running 3' to 5' if you were to look at it from this view. But we typically talk about DNA as being 5' to 3', so the terminology can get kind of confusing. But the thing is, at the 5' end, we have this free phosphate group, and at the 3' end, we have this free OH. Why this matters is that when DNA bases link to, when DNA nucleotides, so Dntps, the like building blocks of DNA, so like our train tracks, when they link together, they do so from the 5' to the 3'. So the DNA polymerase is going to take this incoming Dntp, it's going to use the energy from like those ATP groups from like all that negative charge clamped together to help make this reaction more favorable. But it's going to be building from the 5' to the 3'.
And so in our reaction, when we have these strands running in the opposite directions, it's needing to be building from 5' to 3' in its strand sense. So like we need our primer, we need one of these primers so that this strand is running 5' to 3'. The newly built strand is going to be built 5' to 3', and then this strand is going to be built 5' to 3'. And so this is why we need a primer for each strand. And so in the first one, it's kind of just going to run off, and then it'll keep making copies of these. And we'll look at this in a little more detail in a minute. So a key thing about this is, so this is a DNA-dependent DNA polymerase that we're using. So this means that it needs to use a DNA template to make a copy of the complementary strand. So by complementary, this has to do with the reason why we can do this copying at all is that because the DNA strands, the Dntps, the individual letters, those tracks, they have this specific feature. So all DNA bases, all DNA deoxynucleotides, so have this same generic part. They have this deoxyribose sugar and these phosphate groups, and these are going to allow them to link together to form individual chains. And so they're linked together through these strong covalent bonds called phosphodiester bonds. So these are like strong bonds, which is why we can heat the strands up without disintegrating them. But then between the strands, we can form these things called hydrogen bonds, which are more like partial charge-based attractions. And so these can be broken up when we heat the reaction, whereas when we're heating it, we're not going to be disintegrating the individual strands. But so we can unzip the strands, but because we have these interactions, we can always also zip the strands back so we can get the primers to bind. And importantly, we can use one as a strand for template for making the other, because these pairings, and we can have our primers specifically bind bases because these hydrogen bonds that we're forming are going to be base to base specific. So an A is going to pair with a T, so an adenine and a thymine, and then a guanine and a cytosine, so a G to a C. So these are coming from these unique parts. So all the DNA bases have that same generic part, but then they also have this unique part, this nitrogenous base or this nucleobase, which is going to be one of these four things. And then so it's going to, one will form as like the template track for telling the polymerase to lay down the opposite track. So when we design a PCR reaction, we're going to put in our polymerase, we're going to put in a bunch of those DNTPs, a bunch of our letters. Then we need a couple other things. We need magnesium and like salts and stuff, because basically the DNA backbone is going to be, the DNA backbone has all these like negatively charged phosphate groups. So the magnesium is going to help kind of like shield those charges. And in the post on the DNA polymerase, you can see that the magnesium is actually going to help make this reaction happen and help the polymerase kind of like hold the stuff in place and stabilize the incoming and like the transition state and all of this, because these charge groups are going to interact favorably with those magnesium. The magnesium is kind of going to help the polymerase get a grip on them. So we take our DNA polymerase, we take our DNTPs. Often this is you can get this as like a sort of master mix. So you buy this like pre-mixed, and then you just have to add the stuff that's unique to your reaction. So like you need this stuff for every reaction, but then you want to specify, you're not always going to want to be copying like the same things. So you and the person like down the hall from you probably really doesn't want to be copying the same thing as you do. And so you want to put in your specific template, so the specific DNA that you want copied. So you're putting in that railroad, and then your primers, so those start stations. So we're going to have one per strand. Remember, we need one that's going to be like our forward primer, which is going 5' to 3', and the reverse primer going 5' to 3' on the other strand. So the terminology can get really confusing, and so that's why you need to be really careful when you design your primers that you're using the right like directionality, so you don't tell the, so remember that when you tell the polymerase to start, it's going to be going from the 5', it's going to be extending past this way. So it's going to be extending from 5' to 3', so it's going to be building off of the 3' end of the primer that you put in to keep extending that 3' end, going 5' to 3'. So if you wanted to copy something over here, you're using the wrong primer. So if you want to copy something over here, then that would be, you would have the primer on the other strand so that you're going 5' to 3'. So just be really careful when you're designing your primers. And there are software tools that can help you with this. We'll talk more later about other things to keep in mind when designing primers. So you have your primers and you have your template. Now what's going to happen is, so when you're, we need to start with like the initial start. So initially we have this template that's much longer than the region that we actually want to copy. Then what's going to happen is we need to melt it. So we need to pull those strands apart. And so when we pull we separate those strands, because remember the strands are being held together through all those hydrogen bonding, all that base pairing between the different bases, as well as we saw the other day in my talk on DNA structure, the main driving force for holding the strands together is actually this thing called base stacking. So basically the bases are on top of one another and they're kind of like forming these interactions with one another, like within the same strand and stuff. Because they're all kind of like huddled together, they can kind of like sync up. And so it's more complicated than I want to get into here, but know that it's that like base stacking interactions with like those flat bases on top of one another that are really helping hold this all together. But then the base pairing between the strands are providing the specificity that you need. And so, but all of those are these weak attractions that we can break apart by heating them. So when we're heating, when we heat something, we're adding energy to that thing. This is going to allow it to wiggle more and break free from those attractions. So when we melt, we're going to basically pull these strands will come apart because now these have energy to wiggle apart. Then in the anneal step, we're going to lower the energy, lower the heat, so that the primers can bind. So if at the high temperature, your primers aren't going to be able to bind because they're not going to want to stick. If these don't want to stick, these really don't want to stick because these are going to be shorter. And so the longer your thing is, the more tightly it can bind, and the more energy you need to pull it apart, the more heat you need to pull it apart. So if you have really short things, they're really not going to want to stick when you have a higher temperature. So we need to lower the temperature so that these primers will bind. And this is our anneal step. And then you might need to adjust the anneal step depending on the primers that you use. So depending on like the sequence of the primers, so how like GC rich they are. So if they're more GC rich, they're going to have a higher melting temperature. So you can use a higher temperature. We'll get more into this when I talk about primers in a minute, but just know that you might need to optimize the temperatures of these various steps based on like the primers you're using and that sort of thing. So when you anneal, you're going to lower the temperature so the primers can bind. Then you have an extension step. So typically this is done at a little higher of a temperature, and so that the DNA polymerase has a little bit enough steam to keep moving, but not enough that these primers are going to fall off. And so the DNA polymerase, this first cycle, it's going to like run out of steam or run out of time, or it's going to fall off the end of the strand. Because you're only giving it like a certain amount of time. And so typically you time the cycles according to the speed of the DNA polymerase that you're using. So how fast it can go and how long the thing that you're trying to copy is. And so this is why sometimes PCR can take a really long time and sometimes it can take a really short time, depending on how long the thing you want copied is, because there's cycle time and how many cycles you do. But basically the cycle time is going to depend on how like the thing that you need copied, because it's like if the DNA polymerase is only copying a short little thing, then it doesn't need a whole bunch of time to do that. But if it's a really long thing, you don't want to stop the cycle before, it's only partway through. And so you have to look at like the manufacturer's like information about the different polymerase and how fast they can go and that sort of thing. So some of them are like I think it's like 15 seconds per 1,000 bases or something, is like the one I use. But anyway, so once you do this, what's going to happen? So this first cycle, they're kind of just going to fall off. But then the second cycle, you are now going to use those strands you just made. So you're going to heat them up again. You have another melt step to pull them apart, and your primers are going to bind to those strands you just made. Now what's going to happen is that, so these are the same primers that we had before. So we don't have to, we're not adding anything in between these different steps. We're just like changing the temperature to manipulate when the strands come apart, when the primers bind, that sort of thing. So this time, however, so they're going to start where the primer, they're always going to start where the primers start. So you're dictating that, but now they're going to end where the primer on the other strand started, because they're starting that, because that other strand is now being used as a template strand. So once they get to here, there's no more, and so they're going to fall off. And so you're going to end up, now you have these strands are going to be of a defined length. And so you can do this over and over and over, making lots and lots of copies. And so each cycle, you're going to be doubling the amount of copies you have, because each of these strands that you just made can be used to make another strand. So this is a super useful technique, and we can use it for a variety of things. For example, I have a post on like colony PCR, where basically we're trying to do PCR to figure out if our molecular cloning worked. So if we got like the piece of DNA we meant to get into like a plasmid into the plasmid. We can also use this thing called qPCR, quantitative PCR, where basically we can count the number of copies that get made as they get made. And this is actually the basis of like the PCR tests that you might have heard about for like the coronavirus. But so there's a bunch of different things that we can use PCR for. We can also use PCR when we're doing the cloning, like to make a lot of copies of these parts that we can then like stitch together. So all sorts of different uses of PCR. And so now let's talk a little more about the nitty-gritty details and things you need to think about when you're designing a PCR experiment. So when you're designing a PCR experiment, probably like the main thing that you're thinking about is your primers, because that's what you actually need to do the most work to design. The rest is just like trying to optimize temperature and that sort of thing. But you actually need to design your primers. And so, and when you're doing the designing of the primers, there are various things that you need to keep in mind. So one is that, remember, we're going to be kind of the temperatures that we use are kind of going to depend on the primers. So there's this thing called the TM, or like the melting temperature. It's basically, it's a measure of how tightly those primers are going to bind to the DNA, and kind of like how much heat you need in order to like peel it off. The key thing to keep in mind is, so don't worry about like the actual, you don't really typically have to worry too much about like the actual exact temperatures and stuff that the software predicts. And you might even get different values with different software. But you want them to have a similar TM, so that you don't have like one prime, because you have those two primers, remember, one per strand. You don't want one of them being like bound while the other's just like floating off. And that sort of thing. And so you want them to have similar temperatures. So this is, it matters because so you might think like, well, why don't we just use like a really low temperature and then they'll both bind, right? Well, the thing is that if you have too low of a temperature, what's going to happen is that the DNA can kind of settle for non-perfect base pairing. So remember that it needs energy. When it bonds through those base pairs, it's kind of going to help stick it to the DNA. And we talked about how we add heat to like unstick. So if you have something that's weakly stuck, it's going to take less heat to fall off. If you have something that's strongly stuck, it's going to take more heat to fall off. If you have something that is, so the energy, the heat is going to help determine whether the strands are stuck or not. And then the heat in combination with like how strong the sticking is. So if you have perfect base pairing, you're going to have stronger sticking than if you have like, if there are mismatches in there. And so basically what can happen is that at a low heat, you can get mispairing. So you can have your primers binding to like the wrong places. So binding to places that might be similar in sequence to the target sequence, but not identical. And then they kind of get stuck because they don't have enough energy to wiggle free. Whereas if you have perfect binding, even if you have a higher temperature, you're still able to stay stuck as long as like higher temperature within limits or whatever. And so this is why you want to use like kind of like the highest temperature you can, but you need to make sure that your temperatures of the two are then similar. This will give you the best results typically. So if you have too low of a temperature, you can end up getting the a bunch of different kind of like side products, unwanted products. And so like if you were to run a agarose gel to kind of like check the size of the product, you might see multiple bands and that sort of thing. So you want to, that's going to give you like non-specificity, which you don't want. So in addition to like, you want to make sure that it's only going to, you want to basically minimize also the chances that it's going to form those non-specific interactions. So you want to make sure your primer is unique for the region that you want. So there's software tools like NCBI Blast that you can use to check for other binding matches. So even if you have partial matches, these can lead to non-specific products in multiple bands, as we talked about. And if you're getting these, you can try raising the anneal temp, but that's why it's important that you know like your anneal temperatures and that why you know like the relative TMs of your different primers and that sort of thing. So the chances of having those non-specific interactions are going to be smaller if you have a longer sequence. It's just like statistically, like if you have a longer sequence, it's less likely that something's going to randomly bind. Typically primers are around, depending on what you're using it for, they're typically around like 20, 40 nucleotides, 20 for like, I typically use like 20 for just like a simple type of PCR. If I'm doing something where I need like overhangs for like slick, just the cloning method, then I'll use like 40 and that sort of thing. But thankfully, these primers are pretty cheap, and you can just like order them synthesized and get them in a couple days. In addition to the length, what's going to help determine the like TM is going to be the GC content. So in the most simplistic form, we can think about G and C as having 3 hydrogen bonds, whereas A and T only have 2. And so this is one reason. But as I talked about on a post the other day, the reason why GC-rich regions of DNA are like harder to pull apart is actually because of they have stronger base stacking interactions as opposed to just the GC like base pairing. And so it's more complicated than just having 3 versus 2 bonds. But you should just know that typically the GC content is going to be a major determinant. And so you typically want about 40 to 60% GC content. There's also other things if you're having problems with your primers. There's like different things like people say about like what you want on the ends and that sort of thing. If you're not having problems, you don't have to worry about it too much. But sometimes you do run into problems, then you may need to optimize your primers more. Speaking of things you might need to optimize with your primers, you want to make sure that in addition to making sure that it doesn't bind like other places in the DNA template, you want to make sure that it's not going to bind to copies of itself or of the other primer. So we can call these like primer dimers, and these can lead to when it's like binding to copies of itself or the other primer, and then it can lead to these like little tiny amplified stretches of primer chimeras. And so you might see like little tiny bands on your agarose gel. You also want to make sure that it doesn't like bind back and form hairpins on it with itself, which can block the primer from binding where you want it. And so this is another reason why you don't want to have too low of a melting temperature, is it's going to increase the chances that if you have that you can have something like this if there's a possibility of it happening. So when you're designing primers, there's a bunch of different like software and stuff and tools that you can use. It'll tell you like, oh, those are bad primers. So yeah, so there's software like Blast primer design, various different software tools. So that's the primer. You also may have the opportunity to kind of think about what you want in your PCR polymerase. So typically you use what the lab has, but there are different types of polymerases. And so basically we don't use the polymerase that we used in our body. If we did, it would like melt apart. So we have this issue of thermostability. It needs to be able to withstand the high temperatures of our reaction. And it turns out that the DNA polymerase from extreme thermophiles can. So these are guys that really like it hot. And so thermostability is basically like how hot how much heat can it take before it can break. And so these thermophiles are like these bacteria and archaea and stuff that live in these really, really hot places. So they naturally have heat resistant proteins that we can use. So a common DNA polymerase that's used is like Taq polymerase, like TAQ. But that's not the only thing that you care about. So often we have like modified versions of Taq polymerase that are used because there are other things that we care about. And one of the main things that we care about usually is fidelity. So basically fidelity is talking about like, is it going to make mistakes? And if it makes mistakes, can it fix them? So low fidelity polymerases will make a lot of mistakes, but keep plowing on. Whereas high fidelity polymerases, they can sense the errors and then they can kind of like, they have this proofreading domain. They have this 3' to 5' exonuclease that can kind of like remove the base and keep going. So if you want to make sure that you have a really accurate DNA copying, then you want to use a high fidelity polymerase. But often this is coming at the cost of efficiency, which is like how fast can it go? So you can imagine that if you didn't have to worry about like how accurate it was being, it would just plow really, really quickly. And so it would be very efficient, but not very accurate. So the DNA polymerases that are like commercially used are often, there's often like kind of a compromise between efficiency and fidelity. And you can kind of, there are different versions that you can get based on what you care most about type of thing and how much you're willing to pay. Another thing is processivity. So this has to do with, this is one of the main contributors to efficiency is basically like how far can it go without falling off? So if you have, so basically like it can slip off the tracks and then it has to like find it back again. So often what these like commercially available polymerases, they often have something that to increase processivity by adding something that clamps it onto the DNA. So these can be like separate proteins or like fused together to help it like stay stuck on. So you get higher processivity. And then finally, so often like in the lab stuff, like I care a lot about fidelity. I want to make sure that it's not making a bunch of mistakes. Like I don't care if I have to set my cycler for a little longer, because for the work I'm doing, what I want to do, like I just care about the like I'm doing cloning and stuff. Like I want to make sure that I'm putting in the super duper right instructions into there. But other times fidelity is less important and things like efficiency are more important. And sometimes fidelity is like something you don't want. So they're like, you can get like error prone polymerases where basically they make a bunch of errors as they're going. And this is actually used for things like directed evolution, where you purposely make like random mutations so that you can then be like, then you can see like whether those mutations made like made the molecule, like made a protein that led to a protein that had like a special feature, that sort of thing. So you can do like selection for the ones that are best and stuff. So there are reasons why you would want a low fidelity polymerase. So a high error polymerase. So there's different polymerases that you can get in for your different needs. And there's also different polymerase like mixes you can get. So sometimes when you buy your polymerase, it's like separate, and then you have to add in the Dntps and the salts and everything. Other times you can get like a master mix where basically it has everything in here except for your DNA so your template and your primers that you put in. So So hope that helps you understand PCR, and happy copying.